Hi, dear students. Welcome back to one more session of genetics. And in the previous sessions, we had come across the laws of inheritance, which were being laid out by Mendel. And we had discussed the law of dominance and the law of segregation. And today we would be discussing the law of independent assortment. But before we go into the Mendelian laws remaining, I would like to talk about Reginald C. Punnett, who was a pioneer geneticist from Cambridge University, and he put forward a square called the Punnett Square. Now, what is this and what is the significance of the Punnett Square? The Punnett Square is will help us to devise all the possible genotypes of the male gamete of one parent, and it is written on the one axis of the square, as well as the gametes of the next parent is being written on the second axis, and the possible genotypes which could the offsprings obtain is being put below the squares. Now, if you go to take this particular example, you can see here you have a yellow seed as well as a yellow seed here. This is an example of a hybrid of Mendel's experiments. That's a F1 hybrid in Mendel's experiments, and you can see that the possible gametes which can come here is capital Y and small y. And if you have one more hybrid here, it is capital Y and capital Y. And when this F1 hybrid was being self-fertilized, the possible outcomes or the possible offsprings which can come is capital Y and capital Y, capital Y and small y, capital Y and small y, as well as small y, small y. So the expression of the possible genotypes of the progeny in a checkerboard like box is being explained here, and this box is referred to as the Punnett square. Now this is another representation of the Punnett square which I had explained here, and this diagram is used by the biologist to determine the probability of an offspring having a particular genotype. And you can see that you get a summary of all the possible combinations of the maternal alleles as well as the paternal alleles. And thus what happens, such Punnett squares are used to examine the genotypical outcome probabilities of the offspring of a single trait or when crossing multiple traits from the parents. Now, why I need to say this here, when we are going to consider the next experiments of Mendel, mainly in the dihybrid cross, we'll be considering more than one trait at a time. And so then to express them, it would be easy if we come across or if we are familiar with the Punnett square. It was it was in fact after Mendel had submitted all his experiments that the Punnett square was being discovered. But for the ease of our understanding, I just explained it before the dihybrid cross experiments of Mendel. So now let's go to Mendel's dihybrid cross. You should know that Mendel was not only doing experiments with a single character. After his experiments with single traits, Mendel found that he laid his attention on two or even more pairs of contrasting traits at a time. That is, earlier he was taking only round seeds or yellow color, wrinkled seeds, or each trait was only being taken either round or wrinkled or yellow or green. That was the only way by which he was taking a monohybrid cross. Now, in a second set of experiments, he started taking two contrasting characters together at a time. And a cross involving two character differences separable in inheritance is called a dihybrid cross. In one of his experiments, he crossed a race of peas having yellow round seeds with a variety producing green wrinkled seeds. And he found that in the F1 generation, he got round and yellow seeds. This was a combination. So first he had pure bred pea strains which were round and yellow, round and green, wrinkled and yellow, and wrinkled and green. And when he crossed them, he got round and yellow pea varieties in the F1 generation. And on self-fertilizing them, he got different varieties over here. And using the Punnett Square, we are going to explain that. When the F1 plants were self-fertilized, there were four classes of offspring in the F2 generation. Out of the 556 seeds collected by him, there were 315 yellow round, 108 green round, and there were about 101 yellow wrinkled and 32 green wrinkled, a ratio with is of about 9 is to 3 is to 3 is to 1. And out of these four phenotypic classes, the yellow round and the green wrinkled are the parental types, and the other two types, the yellow wrinkled and the green round, are the newly formed combinations. And based upon these results, Mendel suggested that there could be independent combinations of different pairs of alleles in a cross involving two pair of alleles or more. And this is known as the law of independent of assortment or Mendel's second principle. So when we can now express here the traits which he had taken on a Punnett square, and you can see that the round yellow, the gametes possible are capital RY and capital Y, capital R and small letter r and small letter r and capital letter Y, and the corresponding gametes are also written over here, and they were being crossed. And when it is truly expressed in a Punnett square, and when the information obtained by the experiments were being considered, all the information and the ratios were being found to be significant. And it was noted that the ratio of 9 is to 3 is to 3 is to 1 was being maintained. And thus it was the law of independent assortment was being put forward by Mendel. Now this is known as the second law of inheritance, and it states that a pair of trait segregates independently of another pair during the gamete formation. And as the individual hereditary factors assort independently, different traits get equal opportunity to occur together. And this was stated as the law of independent of assortment of genes put forward by Mendel. Now, something which I would like to tell you about the Mendelian laws is that, of course, we came across the laws which Mendel put forward based upon his experiment, and that was being true for the case of green peas. But you cannot exclusively say that all the laws which he put forward were strictly followed or it was being universally applicable. Now, the law of independent assortment, as for an example, it has no universal application as the phenomenon or the law of independent assortment of alleles occurs only when they reside on different chromosomes. Now, if they are residing on the same chromosome, they are always influenced by linkage, and what happens, they tend to be inherited together and not independently. So, the law of independent assortment of genes is applicable only if the traits are being found on different chromosomes. As well as I would like to also state that the law of dominance is also not being absolutely or strictly followed, as because we can see that there is a phenomenon of co-dominance which is detected in some organisms. So with this, we come to an end of the Mendelian genetics. Hope so you are clear with it. Thank you for now.